Hello, everyone, and welcome to today's live broadcast, Advances in Cellular Automation, presented by Mike Kowalski, Ph.D. We are excited to bring you this educational web seminar presented by labroots.com, the leading social media site for science professionals, and sponsored by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences develops, manufactures, and markets products that simplify, automate, and innovate complex biomedical testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For over 75 years, our products have been making a difference in people's lives by improving the productivity of medical professionals and scientists, supplying critical information for improving patient health, and delivering trusted solutions for research and discovery. To learn more about Beckman Coulter Life Sciences, please visit www.beckman.com. I am Judy O'Rourke of Labroots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window. Type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice you'll be viewing the presentation in the slide window. To enlarge this window, just click on the screen icon located on the lower right. And finally, if you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or you can use the Q&A button to let us know that you're having a problem. Thank you. Now I'd like to introduce Dr. Kowalski. Mike Kowalski is a staff application scientist in the Sample Preparation and Applied Markets Group of Beckman Coulter Life Sciences. He received his Ph.D. in microbiology and molecular genetics from Harvard University. Prior to joining Beckman, Dr. Kowalski was a postdoctoral fellow at the Novartis Institutes for Biomedical Research, where he used automation to screen for novel regulators of stem cell pluripotency and differentiation. Since joining Beckman, Mike has developed automated applications in the areas of cell culture, cellular analysis, and mass spectrometry. Please join me in welcoming Dr. Kowalski. I will now turn the presentation over to him.
All right, thank you, Judy, for the introduction, and thank you, everyone, for attending today. As Judy mentioned, I'll be talking about some of the work we've done to automate cellular applications using our new Biomech I-Series. Before we begin, we do have a few disclosures to show, and we'll now move on to the actual talk itself. So this webinar is part of a series that have been presented, with the earlier talks having described how the Biomech I-Series can provide simplicity, efficiency, adaptability, and reliability. For those who are unable to attend the earlier talks, I'll just give a brief overview of the Biomech I-Series instruments, but mostly we'll focus on how this simplicity, efficiency, adaptability, and reliability can be applied to a variety of cellular workflows, including ELISAs, flow cytometry sample prep, transfection optimization, and basic cell culture. So for the brief introduction, the I-Series come in 2 different formats. The I-5, shown on the left, has a single pod, which can either have a multi-channel head, such as a 96 or a 384 channel head, or it can have a span 8 pipetters, which each of the probes can spread out, allowing you to access multiple tubes at once. Shown on the right is the Biomech I-7. This can have up to 2 heads, so it can have either a single multi-channel or a single span 8 head, or it can have dual multi-channels or one of each, a span 8 and a multi-channel. And it obviously has a larger footprint than the I-5 shown on the left. Some of the new features that are shown on the I-Series include a status light shown at the top of the enclosure to the right. This light can basically indicate what is happening on the system, whether it's currently running, whether it needs to have an interaction, or whether it's in standby mode. And there's also a new deck LED light, which helps illuminate the deck for configuring the deck, as well as just keeping an eye on what's happening. However, this light can be turned off if you're working with light-sensitive reagents. There's also an optional enclosure shown to the right, and this is very useful for cell culture applications because it can be configured with HEPA-filtered fans to ensure a sterile working environment. It also can enable direct integrations, also shown to the right, and we'll cover a number of these throughout the talk, whether it be plate readers, incubators, flow cytometers, etc. And both of the instruments have a configurable high-density deck shown on the lower left. The Biomech i5 deck has up to 25 microtiter plate positions, and the i7 has up to 45 microtiter plate positions. And these can be configured in different ways with different positions for heating and cooling, for trash or washes, etc. And each of the systems features a high-axis rotating gripper. This allows complete coverage of the deck so that all positions can be accessed. And because of the high access, you can also pick up a microtiter plate next to a taller piece of labware, such as a tip box. There's one per head, so if you have two heads on the system, you would have two grippers. When it comes to liquid handlers, obviously liquid handling is still the key, so we have made some improvements to the multi-channel head. One of the main features is that there's now a 1,200 microliter capacity head. This enables you to transfer 1 mil per tip, while also having a 20% additional blowout to ensure all the liquid is removed from the tips. And the head now also features selective tip pipetting, so enabling you to pick up a single column, a single row, or a single tip, or any combination of tips as needed. This obviously gives a lot more flexibility than being forced to always use 96 or 384 tips at once. On the span 8 side, we now have per probe calibration. So obviously we know there may be some slight inherent differences between the pumps, and so this really enables you to dial in the accuracy at the low end as needed. So obviously having the ability to calibrate each probe can really provide some of that additional precision. And also from a simplicity standpoint, we now have a volume splitting feature that all you have to do is check a checkbox, and if you ask to transfer a volume that is more than the tip capacity or the head capacity, the Biomech will do the calculations and split it into the number of transfers needed to reach that volume. We also have a deck observation system. So this basically entails 2 wide-angle cameras, one on each of the front corners of the system. This can enable remote monitoring. Obviously, for longer workflows, you may not necessarily want to stay by the instrument, so this allows you to monitor what's happening on the instrument from your desktop or your laptop computer. And another feature is that it will record 30 seconds on both before and after any error that occurs. So obviously a good way to troubleshoot if something has gone wrong. We also offer the Pro Service Remote Management System, and this can actually allow our tech support folks to access some of the error logs or possibly look at some of those minute-long videos to try and get an idea of what, if any, maintenance is needed for the system. And this should enable them to bring the parts they would need to help save an additional trip. And finally, the system is Windows 10 compatible, which is great for moving forward as more and more instruments will be running on Windows 10. So moving now into the cellular applications, we'll start out with an ELISA method. So I think most people are familiar with ELISAs. It's obviously pretty ubiquitous. It is a fairly labor-intensive process, though, and shown on the lower left-hand side is just the workflow that we use for our proof of principle work. This was an IL-22 ELISA kit that was just commercially available. And the steps include doing a serial dilution using the automation, then adding a diluent and the samples and standards to the plate, shaking it for 2 hours, doing 4 washes, adding a conjugate to the plate, and then shaking for another 2 hours, followed by additional washes, and then a 30-minute incubation with a substrate, adding a stop solution, and then reading absorbance. So a fairly straightforward workflow. But again, one that if done manually can essentially take a good portion of the day and just requires quite a number of liquid transfers. Shown on the lower right-hand side is the deck of the i5. If you wanted to use fresh tips for each transfer, you can see very quickly how the deck fills up, and this is one of the advantages of the high-capacity deck, is that we're able to incorporate all this labware during the workflow. Another one of the challenges for ELISAs is that it's frequently very highly variable in throughput. The same workflow could be used for just assaying 8 to 24 samples, or it could be a much higher throughput type of application where you're screening for IgG-producing clones, where it could be in the hundreds to thousands of samples. So we'll keep this in mind as we talk about more of the workflow. So for the proof of principle, we use the Biomech i5 Span 8 system, kind of shown to the right. And we were able to process a full plate in 6.5 hours. And obviously using the Span 8 system, this would be more of a low to medium throughput, probably 1 to 2 plates a day. Of that 6.5 hour workflow, 4.5 hours were just straight incubations. And obviously when we do ELISAs manually, typically this involves adding wash buffer and then inverting the plate over some waste and shaking it out and blotting it against a pad. This is not something that's very automation friendly. So to ensure that we had removed all the liquid from the plates, we used our optimized pipetting techniques to aspirate around the circumference of the well, sort of at the north, south, east, and west quadrants, and this ensured that we were able to remove all the liquid from the flat bottom wells. We were able to achieve excellent standard curves, as shown by the precision of the reproducibility of our triplicate values on our standard curve, shown on the lower left-hand side, where all the CVs of our replicates were under 5%. And we also had excellent linearity, as indicated by the R-squared value of 0.991 for our standard curve, indicating our automated serial dilution was highly effective. So this is a fairly straightforward application, but obviously if you want to do those types of higher throughput applications that I mentioned earlier, such as clone screening, there are a number of considerations when moving to that higher throughput. The first is that you'll likely need to move to a multi-channel system. The nice thing with having the selective tip feature is that even if you're only assaying 24 or 8 samples, this still can be done using the multi-channel head using the selective tip feature. Another consideration is deck space. As I showed before, this could require a lot of tips, a lot of buffers or reservoirs. That's for a single plate. So if you have additional plates, having the higher deck capacity of an i7 might be valuable. Or you might need to integrate some additional storage positions. And we have labware feeders that would allow you to have lots of tips or plates available for higher throughput. Another consideration is dead volume. Obviously, if you're using a multi-channel reservoir, the dead volume for those are roughly 20 mils, which could obviously be more than you would have available for some of the reagents such as antibodies, but wash buffer, etc., might be very plentiful. And so you would need to consider the balance between speed and throughput versus just how much reagent you have available. And again, using the selective tip feature, you could go ahead and use that for the antibodies while using all 96 channels for the wash. And another consideration is scheduling. Obviously, if you're processing multiple plates and if you want to compare those results across the plates, you need to ensure you have consistent timing. This may not necessarily be so essential for a 2-hour incubation, but some of those substrate incubations at the end of the ELISAs may call for only 5 to 10 minutes, possibly as low as 2 minutes, before you are required to add your stop solution. And for this, you obviously want to put some timing constraints in, and that's one of the nice features of our SAMI EX scheduling software, shown on the lower right-hand corner, is that, again, you can allow the software to try and schedule the plates as efficiently as possible, but for some of those critical steps, you are able to put in constraints and ensure that each plate is treated consistently. So moving on now to flow cytometry sample prep. There are a number of challenges to consider when preparing samples for flow cytometry analysis. The first main consideration is that you require single-cell suspensions. And if you're working with an adherent cell line, this requires a trypsinization step. Now typically, cells may often cluster together during trypsinization, and this is when it's essential to have something like the multi-channel head, where you can do repeated pipetting up and down to ensure that those clumps are broken into the single-cell suspensions that you require. Another consideration is sample throughput, and a lot of this will be determined by your flow cytometer. If your flow cytometer takes single tubes or a carousel of tubes, your throughput will likely be lower than if your cytometer can acquire samples using a plate-based format. However, we do have automated solutions for both of those types of workflows, and in fact, you may be able to process your samples in plates and then do a final transfer to tubes if your cytometer requires it. Another consideration is that most of the workflows for flow cytometry, such as antibody staining workflows, require a centrifugation step to pellet the cells and allowing you to perform reagent exchanges, such as antibody washes, etc. Another consideration is that most of the reagents are fluorescent, and so this typically means they are light sensitive, so you need to consider how long you're able to leave your reagents on the deck of your instrument. And we'll talk about each of these requirements as we go forward. For our proof of principle experiments, we basically wanted to perform a drug discovery screen. And so for this, we used 3 inducers of apoptosis and screened 2 different cancer cell lines to look at their effectiveness, both using dilution curves, so looking at different concentrations of the drugs, and also looking at their effects over multiple time points. We used 1 adherent and 1 suspension cell line. The HCT116 colon carcinoma cells were adherent, and the Jurkat leukemia cell line was our suspension example. As I mentioned before, the HCT116 cells required that repeated pipetting using the multi-channel head during the trypsinization step to ensure we had single cells. We then stained the cells with markers for apoptosis using a fluorescent caspase 3,7 substrate. And we also added a DRAC7 cell impermeable nuclear dye as an indicator of cell death, looking for compromised cell membranes. We then acquired our data using the Cytoflex flow cytometer, and the system we use can acquire 15 parameters and 10 colors, although there are now even better versions of this, I think, that go up to 20 or so colors. And the nice thing about the Cytoflex is that it can accommodate either tubes or plates, and obviously we went with the plate loader for our work today. We then did the analysis using our CalusaG software. We'll now show a brief video that will illustrate some of the early portion of the workflow where we focus on the treatment of the cells.
This video will demonstrate our automated flow cytometry workflow, specifically the initial compound dilution and treatment of our cell culture plate. Shown here, the SPAN8 system has loaded 7 tips and is transferring our drug diluent into a plate that currently has the highest concentration of our compounds in the top row. For this demonstration, we are testing 3 compounds, and so we will have a final dose-response curve of 24 wells each with 8 dilutions of 3 compounds. Following this transfer, one of the new features of the I-Series is having a universal tip type. These tips can then be shared between both the SPAN8 pod and the multi-channel pod, which is very beneficial when only using partial tip boxes. Shown next is the selective tip feature of the multi-channel head, enabling us to load just 3 tips, which will then be used to do a row-wise dilution in a 1-to-1 fashion of our 3 compounds. Once the serial dilution is complete, as mentioned, we will have 24 total wells, 8 different concentrations of 3 compounds. We then load 24 tips using the selective tip feature on the multi-channel head, and these compounds will then be stamped into the cell plate shown in the position behind it. By doing this repeated stamping, not only are we able to do dose-response curves, but we're also able to establish a time course of treatment by just processing partial plates.
So now we'll look at some of the data. Again, this was the HET116 cells. So following the treatment of the cells, we then automated the trypsinization using the same 24 tips in the selective tip fashion, and then added our staining reagents and did our analysis. So looking here, we're looking at a single time point at 48 hours after treatment. And shown on the x-axis is our caspase 3,7 staining, and on the y-axis is our DRAC7 staining. And we basically picked just 3 out of our 8 treatments, sort of representing a high, medium, and low concentration of our compounds. In this particular case, we're looking at 5-fluorouracil treatment. And what you can see is as you decline from the highest concentration to the lowest concentration, the number of cells found in the dead and apoptotic gates decrease, indicating our serial dilution was effective. And in fact, we can actually plot some nice dose-response curves with all of the data that we achieved. And what you can see is that looking at this HET116 cell line at 48 hours, we see that staurosporine and camptothecin were far more effective at killing these cells than 5-fluorouracil, looking at the IC50 concentrations of 63 nanomolar, 317 nanomolars, roughly 1,000-fold more effective than 368 micromolar required to get 50% cell death in this HET116 cell line. So again, a nice illustration of a dose response, but we also wanted to look at different time courses. And shown here is our Jurkat cell line, again the suspension cell line, so no trypsinization required. We simply added our reagents and did our analysis. And this was looking at a single concentration of camptothecin treatment at 78 nanomolar. And what you can see is over time, the untreated cells shown in the viable gate in the lower left quadrant, after 24 hours move into the apoptotic quadrant, so they're staining positive for caspase 3,7, but still very few are staining for DRAC7. And then with an additional 24 and 48-hour treatment, you see more and more percentages of the cells moving into that dead quadrant in the upper right-hand quadrant. This can also be plotted out, and what you can see is a corresponding decrease in the viable cell count at 24 hours with an increase in the apoptotic cells, followed by a subsequent decrease in the apoptotic cells with an increase in the number of dead cells. So again, a nice illustration of time-dependent responses in this particular cell line as shown by flow cytometry. There are some additional considerations when thinking about flow cytometry. We've already touched on some of them. Some of the workflow will be determined again by the flow cytometer if you require tubes or plates. So it's nice to have the SPAN-8 pod with working with tubes. Again, this could be, the entire workflow could be processed in tubes using the SPAN-8, or like I mentioned before, you could actually do the processing in a plate and do a final transfer with the SPAN-8 into the tubes in a very rapid fashion. It's certainly useful to have that multi-channel head for the resuspension of adherent cells if that's required. But again, if folks are working on blood samples, etc., that may be less of a consideration. What we showed today was a relatively simple example of just an add and read type of assay. However, most of the more typical workflows would require antibody staining, and this more of a complex sample prep requiring washes and reagent exchanges. These types of workflows can definitely be aided by integrations, and shown on the lower right-hand side, we did have an i5 system that we demonstrated the integration of the cytoflex itself. So if you were processing large numbers of samples, the integration allows the analysis to begin right at the completion of sample preparation without any intervention. We also, kind of on the right-hand side of that image, you can see a microplate centrifuge that can be directly integrated onto the deck. And again, this is excellent for the spin down and wash steps for those antibody type protocols. We also have the option to integrate a plate washer if this would help assist things. However, the multi-channel can essentially do most of this workflow by itself with the addition and removal of washes, etc. And there's also the ability to integrate incubators, either something small shown as the single plate incubator next to the cytoflex in the image, or much larger, higher capacity incubators such as the Cytomat 2C. And this would be good for, say, long-term staining, or again, just similar treatments like we had, where you would treat cells with compounds, let them incubate, and then bring the cells back up from the incubator and do your staining. Once again, I've already mentioned our SAMEX software. This software would also be useful for this type of application for these higher throughput methods, where again, it can process multiple plates in staggered fashion. And once again, you can put in those type of timing constraints for the steps that are essential. One example would be trypsinization. Obviously, you want to consistently treat your plates, because if you have longer trypsinization times, this could actually change the cell surface expression of some proteins as more of them get cleaved during longer trypsinization treatments. So we'll now move on to another example, being transfection optimization. So when I think of transfection, it seems relatively simple. Essentially, it's just combining a lipid with a nucleic acid and putting it on top of cells. However, this it's not necessarily simple for every cell type. And because of this, an optimization approach can help. Unfortunately, optimizing transfection conditions is not so simple. For this example, we chose to use a design of experiment, or a DOE approach, where we changed the different lipids we were using. We tried different lipid concentrations, different concentrations of nucleic acids, and different cell numbers to see if we could identify an optimal condition for a variety of different cell lines. The problem with this approach, or the difficulty with this approach, is that first off, from an automation perspective, you need ways to create these combinations. And again, this is a nice example of how having the selective tip feature for the multi-channel head really enables some of these, because you can do row-wise or column-wise transfers, as well as any combination you might need. In addition, the other problem is that the conditions increase factorially. So it's very quickly, as you add more and more conditions, the number of samples you need to test grow pretty dramatically very quickly. And we'll show an illustration of that as well. So the workflow the approach that we took to automating the transfection optimization is we started with an empty plate and then tried 6 different lipid conditions, really 5 different commercial lipids that were available, Darmafect 1 through 4 and lipofectamine, and one condition that had no lipids, so essentially our untransfected control. We used our SPAN8 system to transfer these reagents from tubes into the upper row of the 96-well plate, and then also used the SPAN8 to just fill in the next 18 wells with media for serial dilutions. Shown in the dark and light purple, we also used the SPAN8 to transfer 2 different concentrations of our nucleic acid. We did an siRNA control. And on the next step, we loaded 6 tips on our multi-channel selective tip, much like you saw earlier with the 3 tips for the flow cytometry example. And we did a serial dilution in a row-wise fashion to get 24 different conditions of lipids, and then stamp those into the lower 24 wells using the same 6 tips. We then used our SPAN8 at the same time to essentially stamp out those nucleic acids into the remaining 48 wells on the right-hand side. We then loaded 48 tips on the multi-channel selective tip and combined those 48 wells on the left to the 48 wells on the right, resulting in 48 different conditions of nucleic acid and lipid combinations. Following an incubation to allow for transfection complexes to form, we then stamped those 48 conditions into a 384-well plate and then loaded 96 tips to transfer 2 different concentrations of cells, resulting in 15,000 or 7,500 cells per well. The final result was 96 different conditions, and shown in the plate map below, and the 4 blue wells indicate the 4 replicate conditions for a given condition, such in this case was 0.8 microliters of Darmafect-1 with 100 nanomolars of siRNA and 15,000 cells per well. So the nice feature here is we're able to test 96 different conditions in quadruplicate for optimization in a single plate. If we tried to do this manually, my best estimate would be that it would take about 138 manual transfers, and instead the automation was able to achieve this in under 20 minutes. We then tested this approach on 3 different cell lines, the HET116 cells I've already mentioned before, and 2 additional cancer cell lines, an ACHN line, which is a renal carcinoma, and a PANC1 pancreatic carcinoma cell line. We used SI-Glow green as our transfection marker. This is a fluorescently labeled siRNA. And 24 hours after transfection, we added our same DRAC7 reagent as a marker for toxicity. And we then performed a media exchange using the multi-channel head again to try and reduce the amount of background we got from the SI-Glow green. We then did some imaging with the SpectraMax i3X with Minimax 300 imaging cytometer shown in the upper right. And this basically enables 4x images to be taken in bright field and 2 fluorescent wavelengths, 541 nanometers and 713 nanometers. And what this enabled us to do was to count total cells and then get an idea of our transfection efficiency by counting the number of green cells, indicating our SI-Glow green had been taken up. We also looked at toxicity, so the number of red nuclei divided by the total number of cells. We looked at whether we had either inhibited cell growth or whether we had lost any cells during our media replacement by comparing our transfected wells to our untransfected wells in the bright field images. And then we also looked at transfection consistency by looking at the CVs across our quadruplicate wells. So no need to really look at the numbers here, but I'll basically summarize. The first set of wells on the left, green is good. This is basically an indication of getting over 30% transfection. The next set of columns, red is bad. This is basically any conditions that resulted in toxicity more than 5% above control wells. The next set, brown is bad. It's kind of difficult to see. But again, this is where we had less than 80% of our untransfected cell counts in a well. And then in the final set of columns, light green is good. This is where our transfection CVs were below 10%. So again, really the take-home message here is we sort of identified the best conditions by the bold squares around those wells. And the take-home here is that you can definitely see there are differences across the 3 cell lines. And we'll really identify those in the next slide. So shown here are kind of the resulting optimal conditions for each of the cell lines. And again, what you can quickly see is that there are differences between the cell lines. All 3 of them had different ideal lipids that were used at different concentrations of siRNA. But through optimization, we were able to achieve roughly 50% transfection efficiency with all 3 cell lines with acceptable levels of toxicity and with CVs below 10%. Some of the cell lines like PANC1 and HET116 were relatively transfection friendly in that there were a couple different concentrations that might work, but the ACHN cells were significantly different and really more troublesome to transfect. And really, we did have to find that optimal condition to achieve that type of transfection efficiency. Some of the important take-home messages were that even a simple change, even just a twofold change in lipid concentrations, you would see very dramatic effects. Twofold too low and you would see very low transfection efficiency, basically nothing. Twofold too high and you would see rampant toxicity depending on your cell line. So again, really driving home the need to do this type of optimization approach. In addition, because we were able to work in 384 well format, this helped reduce our reagent usage. So again, these are not cheap reagents, so obviously the more you can do in a smaller volume, the better. But to try and do this manually, this really adds challenge to this because, again, it's very difficult to tell which wells you've added reagents to, etc. And another nice thing is now that we have this automated approach, it's very easily adapted to different either nucleic acids or different assays. So if you wanted to adapt this to an overexpression type of assay, just change the reagents, possibly change your transfection reagents and add plasmids instead of siRNAs, and it should essentially work. Alternatively, if you didn't want to use a fluorescent siRNA as a marker, instead you had some sort of phenotypic readout, it might just be requiring changing the assay approach at the end. So now we'll sort of conclude with basic cell culture approaches. And again, this is something that not necessarily everyone thinks about automating because it's something that was typically done at the bench, but this really is something that can benefit from automation. There's a number of reasons for that because there are a number of challenges associated with cell culture. The first is that it's basically very time consuming, and this can be because it recurs frequently. You're probably passaging cells 2 to 3 times a week. If you have multiple cell lines going at the same time, the throughput can become really burdensome, and it's relatively inflexible. Cells don't really care if you have an all-day meeting scheduled. If they're confluent, they need to be passed that day. In addition, there's high variability in this type of work. For example, sometimes you just have cells that you're waiting to plate later in the week, and so maybe you don't have that much work, but Monday may look very different than Thursday, or this week may look very different than next week. In addition, we know there are people in the lab who passage cells differently than I do. And so, for example, we all sort of come up with our own approach for passaging cells, and it sort of just depends on how we were taught or maybe what the written protocol says. But we know there's inherent variability in the way people do things. And so by turning to automation, this variability can go away because a lot of the times this variability will show up in your future assays. And finally, it comes down to scheduling as well. When do I need cells? When am I going to plate my experiments? When I'm doing multi-day workflows, frequently, again, some of these cell culture assays may take days to weeks. What planning ahead do I need to do to ensure that I'm not here all day on a weekend? And so these are just some of the considerations that can hopefully be addressed through automation. For the work we've done, this will be done on an i7, same as most of the work has been done previously. And it features a number of integrations, which we'll show shortly. The work we did involved just culturing 2 adherent cell lines, CHO cells, which we'll show most of the data for, as well as HET116, and a suspension cell line, the JERCAT cells. Basically, we were passaging the cells in 6 well plates, so essentially larger well plates to have enough cells to plate for experiments. We would pass a known cell number every 2 to 3 days, and then we would plate these cells in either 96 or 384 well plates for future assays as a proof of principle. The system itself utilized an integrated Cytomat 2C incubator to allow us to culture our cells directly and bring them onto the system in an automated fashion. It also had an integrated BiCell XR cell viability analyzer shown on the right, and this would enable us to count our cells at time of passaging. It also had a tilting out for larger well plates to basically allow the media to slide all to one side and enable us to remove that media without disturbing the cells. And we also had the integrated SpectraMax i3X with Minimax that I mentioned earlier, and we used this for some imaging and cell counting assays, which we'll describe later. So now we'll show a video again that will really demonstrate how this workflow proceeds and how we utilize the different integrated instruments.
So shown here is the automated approach we took to automating cell culture, starting from the larger 6 well plates and moving assays into 96 well formats. Shown here is the tilting alp, and the advantage of this is that the media can be removed from the edge of the well without disturbing the adherent cells on the well. We use the span 8 system with a P1000 to reach into the corner well and remove the media, and then another P1000 tip to dispense 800 microliters of trypsin to remove the cells from the surface of the plate. This plate was then incubated at 37 degrees for 5 minutes, and this was done on a static Peltier, but could easily have been done on the integrated incubator as well. We then used our optimized pipetting to pipette around the circumference of the well to make sure the cells were removed from the surface of the plate, and then also did a mixing step to ensure that the cells were distributed into a single cell suspension. A portion of these resuspended cells were then added to the sample cup for the integrated ViCell cell viability analyzer, and the ViCell returned a viable cells per mil number, which the biomech was then automatically able to use to calculate what volume of cells needed to be transferred to reach a certain number of cells. If the volume was over 1 mil, the capacity of the tip, the split volume step could be used to automatically transfer the larger volume. In this case, we dispensed 1 million cells into the subsequent well for continuing passages, and a second subset of cells was then added to a large multi-channel reservoir containing 100 mils of media. One of the advantages of the large capacity multi-channel head is that we are now able to completely mix these large volume reservoirs and ensure the cells were evenly distributed throughout the reservoir. In addition, we were able to increase the throughput by using a multi-dispense feature, allowing us to plate 4 96 well plates with 200 microliters per well in a single transfer step. Again, this can be used to increase the throughput and process numerous plates simultaneously or process as many plates in subsequent fashion as needed.
So hopefully that gives you a pretty good idea of what the automation looks like and what the workflow looks like. We'll now look at some of the data that came off the system. So the first question is just regarding the passaging from one 6 well to another. So all the work we'll show here was done using the Chinese hamster ovary cells grown in adherent conditions. And so demonstrated here is basically the results of one of our 6 well passages. Shown below is basically taking 4 regions of interest and taking 12 images for each region using the SpectraMax Minimax imaging cytometer 24 hours after plating. And what you can see is that basically there's no clumps of cells, no clusters, so it looks like we had a really effective trypsinization and mixing, and also it looks very evenly distributed across the 4 regions. So what you can see on the left is what I could achieve manually, and on the right is what the system was able to do in an automated fashion. We got very comparable percentages of confluence, right around 60%, and very low CVs in both cases, indicating, again, good dispersion across the wells. Looking across multiple passages now, so shown here are basically the automated and a comparison in manual of what I was able to achieve. And we look at, on the left, at cell counts and looking basically at how consistent those were across passages and also across automation versus manual. And on the right-hand side is plotting viability, and so we were able to achieve consistently above 90% viability in all 6 of our automated passages. More interesting is that actually, if you look, we were actually able to achieve results that were more consistent than I was able to achieve manually. So again, looking at CVs of 7.9% in an automated fashion when comparing cell counts across 6 passages was better than the 13.8% CVs I was able to achieve manually. You'll also notice that there was slightly less cells on average in the automated well, and that's likely because when we have the plate in a tilted fashion, I can't actually angle the pipette the way I can when I'm performing it manually. So there's that small 5 to 10%, probably 5% of the well that may not necessarily get rinsed down during the automated passage. So we're likely plating the same number of cells and culturing the same number of cells, but can't quite harvest the same number of cells. But again, we can do this more consistently, and this consistency can hopefully lead to more consistent results with our assays. When it comes to actually plating, the video demonstrated plating into 4 96 well plates, but we actually decided to take it one step further and try and plate into 2 384 well plates doing 100 microliters multi-dispenses, much like we demonstrated in the video. And so again, 100 microliters per well times 768 wells. And what we were able to see is that we did, again, imaging after 24 hours. And we then looked to see, did we get consistent distribution of the cells across the plates? So essentially, that did our mixing in the reservoir work? Did we get roughly equivalent cells in all regions of the plate? And that's really 8.2% or lower across all the columns and all the rows across both of the plates. And finally, we looked at how consistent was our multi-dispense. Basically, were we getting the same number of cells for each dispense in all of the 8 quadrants? And when we compare the average counts per quadrant, we saw CVs less than 7%. So again, a very consistent passaging and plating, hopefully resulting in consistent assay results. Another consideration that we touched on earlier is multi-day workflows. So for example, we've just plated our 96 or 384 well plates. How long do these plates need to incubate? Is there an additional treatment? How long till we assay them? These things all have to be taken into consideration because very rarely is this a single day of approach. So when automating these types of approaches, there's a number of requirements. One main one is that you need to have good resource management and scheduling. And this comes back to our SAMI EX software, where again, when you're processing multiple plates, it will find the most efficient way to process those plates in batch-wise fashion. It also requires sample tracking. You need to know where your samples are at any given time in the workflow. And you also need to know if your biomech will be available on a given day. So if you have interleaved multi-day workflows, our SAMI process management software can help schedule these things on a calendar so you can ensure that, again, will you need to come in on a weekend? Is the instrument already being taken up for other assays on a given day? And finally, you need good data tracking, and this is where our data acquisition and reporting tool, DART, comes into account. Basically, these types of multi-day assays may be made up of little islands of automation, right, where you will bring it in on day 0, seed the cells, send them to the incubator, bring them back on day 3, treat them. DART is basically our tool that every time that plate comes in, it can read the barcode and know everything that's been done to that plate to date. And all of that information is carried forward so that eventually your treatment and your eventual assay result will all be located in the same database on the same well. So to wrap up the automated cell culture portion of the work of the talk today, hopefully I've shown you that we were able to handle larger well plates and flasks, really using the span capabilities. But having that combination of pipetters, the span for those 6 well plates and the multi-channel for the micro-titer plates, really is valuable for this type of workflow. There are some caveats. One is that you may need to do some optimization for any given new cell line. One example is the frequency of passaging. So for example, when we were passing the JERCAD cells, we would pass them every 2 days and they were completely happy. The CHO cells, when we tried to passage them every 2 days, seemed to actually die off a little bit, and so that's why we backed it off to 3 days. Another consideration is the trypsinization workflows for adherent cells. One is just the trypsinization time. 5 minutes of incubation worked great for the CHO cell lines, but the HCT116 cells were a little more aggregated, and so you might want to consider extending that time closer to 7 to 8 minutes. In addition, the volume that you transfer into the next well is somewhat dependent on just your cell size. For example, if you have a confluent HCT116 well, you can fit about 5 million cells per well, as opposed to the CHO cells where a confluent well only had 2.5 times 10 to the 6th. So if you were transferring 1 million cells into the next well and you were concerned about residual trypsin, you're actually transferring twice the amount of volume in the CHO cells as you are on the HCT116. So if you do find the need to either spin out any of those reagents, obviously there are some automated solutions for that as well, and you can do media exchanges and not have to worry about residual trypsin. And finally, it comes down to mix steps as well. As I mentioned, the HCT116 cells were a little more clumping than the CHO cells were, and so we basically increased the number of mixes that we performed. Another consideration is the plating needs. One of the nice features of the biomech is that you're able to do data-driven decisions. So for example, if your cells haven't grown as much as you thought they might and you're already in the middle of passaging them, you can actually just have some if-then statements indicating that if you don't have a high enough number of cells to do your cell plating, you can just go ahead and passage them and push that off for another couple days without wasting all the plastic. So that basically wraps up the talk. We'll kind of try and bring it back to the simplicity, efficiency, adaptability, and reliability, and see if we've hopefully shown some examples of how these apply to cellular applications. When it comes to simplicity, having that volume splitting feature, where again, it really doesn't necessarily care what the volume is you need transferred. You don't necessarily need to put in any sort of conditional statements or scripting. Whatever the results from the bi-cell come back, we can always just basically have it split the number of transfers it needs to get the correct volume. Also having the integrations be more direct. Obviously you can fit a lot of instruments in a smaller footprint and not necessarily requiring any additional robotic transports to access these devices really increases the simplicity of those types of integrated systems. When it comes to efficiency, we have the high capacity deck. There's just more room, more real estate to work with, so you can really have these more highly complex workflows as you need. Can all fit on potentially onto the same deck. And there's also the ability to do the large volume transfers with the multi-channel head, as we demonstrated for the plating video. And obviously this can really just make it a more efficient workflow. You can process more plates in a more rapid fashion. When it comes to adaptability, hopefully I've shown that we were able to automate a pretty diverse number of workflows on the same system, really kind of indicating that, yes, these really can be adapted to pretty much any need you have. And also the ability to use the multi-channel selective tip really does increase the flexibility of that multi-channel head. So again, you're able to do more with that head alone. And when it comes to reliability, obviously having the remote monitoring, some of these cellular applications, the ELISA in particular, can take multiple hours. So if you want to just be able to check on your system, it should still be running, but again, being able to monitor what's happening remotely does provide you that extra sense of security. And having our pro service, being able to access any sort of error logs or those video files, really should be able to help decrease any downtime that the system might have. So with that, I'll wrap up. Obviously, we have more information available. I've just kind of touched on some of the applications, but if you want more details, such as the volumes we were using or the specific reagents, that type of information can be found in our application notes, as well as a variety of other resources that can be found at biomech.beckman.com. And with that, I'm happy to take any questions.
Thank you, Dr. Kowalski, for that informative presentation. So it is time for Q&A. If you have a question you'd like to ask Dr. Kowalski, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. And our first question is, have you successfully automated cell culture without the tilting out by optimizing pipetting?
That's a great question, and again, it might necessarily depend on a given workflow. For example, the large one-well plates, as they're called, that would potentially be a little trickier to kind of work our way around, but the 6-well plate, we are able to aspirate around the circumference, and that would be just as effective way of removing the media without disturbing the cells. It might just take a little longer to kind of execute that process. So there's definitely a time and a place for the tilting out where it provides value, but certainly not required for most of the plates up to the very large, maybe one-well plates, or for example, the auto flasks, which really only have a single point of entry. That's where, again, the tilting out might be required. Otherwise, it may just have benefits for some of the plates, but probably isn't necessary.
How hard is it to adapt the automation to different workflows?
That's also a good question. So the workflows I've shown today were all essentially done using the same deck, but obviously it depends what system you'd be working on. Having the i7 hybrid sort of provides you with the most flexibility, but there are certain occasions where, again, depending on your workflows, if things are changing dramatically, you can imagine possibly needing to change what devices are integrated to the system. One of the nice things on the iSeries is, again, there's more universality of the integration, so those do become more easily swapped in and out. But when it comes to just changing the positions on the deck, for example, if you needed more heating and cooling positions or more shaking positions, or had some direct integrations that changed your deck configuration, that's very easily done. That's basically just popping out the current alt positions, putting on the next positions, and then doing a quick auto frame using our tools that are available. When it comes to possibly swapping out heads, for example, you can change between the 96 and the 384 multi-channel head. That process is basically just unscrewing 4 screws and screwing them back in. So even changing for that type of workflow is very rapid. So it's basically built and designed to have high levels of flexibility. So again, barring some sort of massive reconstruction of your needs, it should be very quickly able to switch between workflows, or otherwise you may be able to just automate a number of workflows on a single configuration.
How do you handle contamination?
Yeah, so this comes up a lot actually. So in 7 years of doing this, I've only run into it once, and my way of dealing with it was to throw out the contaminated plate. I really didn't have any other problems. That's kind of one of the benefits that, in theory, if things are working right, you shouldn't really be spreading contamination. So if you want to treat it the way I would treat a contaminated incubator, you can spray things down with ethanol, but just throwing away the plate was sufficient for me. I didn't really have any continuing problems after that. There was not really any need to do anything extreme. But there are certainly, we do have information on chemical compatibility if people really need more information about sterilization. But honestly, for me, everything stays pretty well contained within the plate, so throwing it out took care of my problem.
And as we're waiting for additional questions to come in, I would like to once again thank Dr. Kowalski for his presentation. Do you have any final comments?
Again, just want to thank everyone for attending today. Obviously, we're available for additional information, and when it comes to cellular workflows, or obviously my colleagues have presented on some genomic workflows as well. So this is obviously just one aspect of what you can do with automation and what you can do with the Biomech I-Series. So we're happy to always provide more additional information as required.
Thank you. I would also like to thank our sponsor, Beckman Coulter Life Sciences, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through October 20th, 2017. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.